All right, let's get right into it. You've probably heard people talk about good cholesterol and bad cholesterol. Well, today we're putting the spotlight on the hero of that story, high-density lipoprotein, or HDL. So what exactly does it do to earn that good guy reputation?
So that's the big question, right? What is this heroic mission HDL is on? Why is it so incredibly important for keeping our hearts and arteries healthy? The answer? Well, it lies in a process that is just as elegant as it is essential. And here we have it. The mission is called reverse cholesterol transport. This is HDL's entire reason for being. Honestly, you can think of it as the body's personal cleanup crew. It travels to the most remote parts of our tissues, collects all the extra cholesterol lying around, and hauls it all the way back to the liver to be thrown out. This exact mission is what keeps our arteries clear.
Okay, so here's the game plan for this explainer. We're going to start with what makes HDL so special. Then we'll follow it on its scavenger hunt, step by step. We'll meet the key molecular players that help it out, see how it's connected to heart disease, and then wrap it all up with a quick sheet to make sure it all sticks.
First things first, let's really get to know our hero particle. What exactly is HDL made of, and how is its structure perfectly designed for the job it has to do?
So check out these characteristics. They really tell the whole story. HDL is the smallest and, you guessed it, the most dense of all the lipoproteins. Why? Because it is jam-packed with protein. We're talking about 40% of its entire mass. And that high protein content is the secret sauce for its main job, reverse cholesterol transport. Its journey, as you might expect, kicks off in the liver.
Now, if we were to crack one of these HDL particles open, here's what we'd find. At the very center is a hydrophobic core, safely tucked away, and it's full of something called cholesterol esters. That's the packaged-up cholesterol it's carrying. And wrapped around that core is a shell that loves water. It's made of phospholipids, some free cholesterol, and these special proteins called apoproteins, especially one called ApoA-I. This brilliant two-part design is what lets it cruise through our bloodstream with no problem at all.
And this number you're seeing, 1.063 to 1.21 grams per milliliter, that's its density. This really high number is a direct result of that massive protein-to-lipid ratio we just mentioned. I mean, it's literally what puts the high density in high-density lipoprotein. It's built to be a compact, super-efficient cholesterol shuttle.
All right, now for the fun part. Let's actually follow HDL on its mission. We're going to trace its journey through the body as it plays the role of a microscopic scavenger, cleaning everything up.
You know, every hero needs an origin story, and HDL starts right here. It's born in the liver and intestine, but it doesn't look like a sphere yet. It starts out as a flat, disc-shaped particle called nascent HDL. Think of it as an empty cargo ship, just equipped with the essentials, phospholipids, and that all-important ApoA-I protein, ready to head out and start picking up shipments.
The mission itself happens in 4 key stages. First, it's got to collect the cholesterol. Second, something called esterification happens, which is really just a chemical trick to trap the cholesterol inside. Third, it can actually swap cargo with other lipoproteins. And finally, the big drop-off back at the liver. Let's look at how it grows up.
So this is the transformation sequence. As that flat baby HDL particle cruises around and starts grabbing free cholesterol from our cells, it starts to fill up. And as it fills, it swells from that flat disc shape into the mature round sphere we recognize as HDL. It's literally getting plump with all the cholesterol it's cleaning up.
Now, HDL is a hero, but it doesn't work alone. It's got a whole team of molecular sidekicks, enzymes, and other proteins that are absolutely crucial for the mission's success. So let's meet the cast.
Okay, let's run through the lineup. First, we have ApoA-I. This is the team captain. It provides the structure, but it also flips the on switch for our next player, LCAT. LCAT is the enzyme that actually does the packaging. It takes that free cholesterol and esterifies it, trapping it in the HDL core so it can't escape. Now, CETP is super interesting. It's like a trader. It swaps HDL's packaged cholesterol for triglycerides from other lipoproteins like VLDL or LDL. It's all about making the entire lipid transport system run smoothly. Finally, there's ApoC2. Think of this one as a helpful tool that HDL can lend out to other particles to help them do their jobs.
And this right here highlights the single most important partnership in this whole story. ApoA-I isn't just along for the ride. It's the trigger that activates LCAT. Without the signal from ApoA-I, LCAT just sits there, and the cholesterol can't be properly trapped inside the HDL particle. This makes ApoA-I the absolute linchpin of the entire process.
Okay, so let's zoom out from the molecular level and see why this all matters in the real world, for real patients. This is where we see the direct link between HDL's mission and our risk for heart disease.
When you look at it this way, the clinical picture becomes crystal clear. High HDL levels, that's great news. It means your body's cleanup crew is working efficiently, which is highly protective for your heart. But on the flip side, low HDL is a major warning sign. It tells us that the reverse cholesterol transport system isn't working well, and that's a huge risk factor for clogged arteries and coronary heart disease.
And if you want to see what a complete system failure looks like, look no further than Tangier disease. It's a rare genetic disorder where people have basically zero HDL in their blood. As you can imagine, reverse cholesterol transport comes to a screeching halt. All that cholesterol builds up in the tissues, famously causing large orange-colored tonsils. And sadly, these patients are at a much, much higher risk for developing heart disease at a very young age.
All right, let's boil all of this down into a high-yield summary. If you're studying for an exam, these are the absolute must-know points.
So here's the entire story in 4 bullet points. 1. HDL is good because of one thing: reverse cholesterol transport. 2. It starts as a flat nascent particle and matures by picking up cholesterol, which gets locked inside by the enzyme LCAT. 3. This whole process protects your heart. And 4. Low HDL equals high risk. It's as simple as that.
Okay, time for a quick pop quiz. Let's see what you remember. Which enzyme turned on by ApoA-I is responsible for packaging up that cholesterol inside the HDL particle? That's going to be LCAT, Lecithin-Cholesterol Acyltransferase.
And one more for you. What do we call that genetic disorder where HDL is almost non-existent, causing cholesterol to pile up in the tissues? You got it. That's Tangier disease.
So, let's wrap up with this final thought. Your HDL level is so much more than just a number on a lab report. It's really a performance review of your body's most important cleanup service. It's a story of transport, transformation, and protection, and it makes HDL a true main character in the ongoing drama of your cardiovascular health.